Generally, this is a topic that most of the students struggle with. But I am sure after looking at it the way I'll show it to you, it will be easy for you to understand, remember and answer questions based on glycolysis, Krebs cycle and electron transport chain. Alright? Like I always say, purpose, design, function, PDF will save us today. Now, before I go into elaborating the details of the three processes, three metabolic pathways, let us look at it this way. Where are these three things happening? In a cell, it happens in the cell. All the cells will have all these three happening. We are talking about eukaryotic cells. In prokaryotic cells, the mechanism continues to happen. Even in the prokaryotic cells, the mechanism will happen. The process will happen. But the location will be different. See, the one simple thing that you have to understand between prokaryotes and eukaryotes is, which is the energy producing factory of the cell? Which is the ATP generating factory of the cell? Mitochondria. Do prokaryotes have mitochondria? Then do they not produce ATP? Do prokaryotes not produce ATP? They do. How? There is no mitochondria, but how are they producing ATP? Huh? What? Mesosomes. Okay. Mesosomes are not for producing ATP. Okay, anyways. There are photosynthetic prokaryotes, photosynthetic bacteria, cyanobacteria. Where is photosynthesis happening? In the chloroplasts. They don't have chloroplasts. So where is, how are they doing photosynthesis? Is mesosome doing that also? How? I'll give you an example. Let us assume this is a big classroom. I told you this. In an earlier class also I told you this. This is a big classroom. There are 10 classes going on, 1st standard to 10th standard. All the students of every class is there in the classroom. Teachers of the respective classes are also there in the classroom. The teachers are teaching their respective topics. The students are listening to the respective teacher. If it is one room in which all of this is happening, will it be efficient? Two things. One, teaching is happening. All the teachers are teaching what they are supposed to teach. All the students are trying to listen to the respective teacher that they are supposed to listen to. But it will not be effective because there is a lot of clutter. There is no focused attention of either the teacher or the student. Though the teaching and learning process is happening, it is not efficient. You understand? That is, but if we convert this entire big space into 10 rooms, and each room is dedicated for one class. It is the same number of students, it is the same number of teachers, it is the same teaching, it is the same listening. But teaching will be more effective, learning will be more effective, and the focus will be better. Outcomes will be better. What did we do? Instead of everything happening in a common space, we compartmentalized focused groups and isolated the focused groups from each other. You understand? So one big classroom with all the classes, all the students, all the teachers being present is a prokaryotic cell. Whether it is glycolysis, Krebs cycle, electron transport chain or photosynthetic reactions, all of this is happening with something to start with, something to end with, something to mediate. All are chemicals only, no? There is a starting chemical, there is an end chemical product, there is a substrate product. The starting chemical is the substrate, the end chemical is the product, something that mediates is the enzyme. All are chemicals. What happens in a prokaryotic cell? In a prokaryotic cell, the substrate is also there, the enzymes are also there, products are also being formed, but the efficiency is low because they are not concentrated in focused clusters. In eukaryotic cell, function specific enzymes purpose specific enzymes are clustered into compartments. So chloroplast is able to perform photosynthesis better because all the enzymes required for performing photosynthesis are clustered together, isolated with a membrane, so that it doesn't get influenced or there is no interference from something else happening. Alright, that's all. So mitochondria is also performing ATP synthesis with the help of enzymes. The enzymes are present in prokaryotes also. Prokaryotic reactions, energy production or photosynthesis, whatever is the reaction that is happening in the cytoplasm. Sometimes, are mesosomes permanent structures? Are mesosomes permanent structures in bacteria? They are temporary folds of membrane. So it's like, to improve the efficiency of a particular process, the bacteria can bring together a set of enzymes and enclose it with a temporary membrane to carry on a particular process. Mesosome is a temporary structure. It's not an organelle. But can you see that from being completely cytoplasm dependent reactions to bringing about some adaptation where it can bring together certain enzymes and package it into a temporary membrane, to moving into a structure where everything is packaged into permanent isolated clusters, organelles. See the evolution? Alright? So everything is happening in every living organism. Everything is the same. How it happens, where it happens, to what extent it happens is the difference. Remember the first discussions that we had. Survival, reproduction, energy dynamics, matter transformation, transformation of matter, how one substance is being transformed into another substance, the elemental interplay, the combination, recombination of different elements in different proportions bringing about production of different chemicals. That's all, no? Life is so simple. You have elements in nature, combination of elements in different proportions will give rise to certain chemicals. Those chemicals will be acted upon by enzymes to produce something else. What are enzymes doing? Enzymes are restructuring. Whatever the enzymes act on, they will restructure it. They will restructure it into something else. It can be bigger than what it acted upon or smaller than what it acted upon. If the enzymes are making smaller products, you call that reaction as a catabolic reaction breakdown. Enzymes are facilitating formation of bigger products, you call it anabolic reaction. All the chemical reactions happening in living systems, you call it metabolism. That's all. Alright? This is also metabolic process. Glycolysis, Krebs cycle and electron transport chain is also metabolic process. Now that you know that these things will happen in prokaryotes also, we will come back to eukaryotes. What happens? Where does each of these happen? Where in a cell does glycolysis happen? By glycolysis, I mean breakdown of glucose to pyruvate. Where is it happening? In the cytoplasm. It happens in the cytoplasm. Then where is the oxidative decarboxylation happening? Pyruvate to acetyl-CoA. Where is it happening? Pyruvic acid to acetyl-CoA formation. Huh? It is happening in the mitochondrial matrix. Can it not happen in the cytoplasm? It can happen in the cytoplasm also because acetyl-CoA is not only specific to glucose metabolism. Acetyl-CoA is a common chemical which is participating in metabolism, protein metabolism, glucose metabolism or carbohydrate metabolism. So acetyl-CoA can get into lipid synthesis process. Acetyl-CoA can get into protein synthesis process, amino acid synthesis process. It's all interconnected. Something is formed, it need not go into the same pathway, it can go into something else. So we'll limit it to glucose metabolisms right now. So acetyl-CoA, pyruvate to acetyl-CoA is happening in the mitochondrial matrix when we are looking at the glucose metabolism pathway. Pyruvate enters the mitochondrial matrix, it is converted into acetyl-CoA by oxidative decarboxylation. Where is Krebs cycle running? Where is Krebs cycle going on? Krebs cycle is happening in where? Where is Krebs cycle happening? It's happening in the mitochondrial matrix. Krebs cycle happens in the mitochondrial matrix. Where is the electron transport chain present? Where is the electron transport chain present? Electron transport chain is a set of complexes embedded in the inner mitochondrial membrane. All of you know that mitochondria is a double membrane structure. There is an outer membrane, inner membrane. What is the space between the two membranes called? What do you call the space between the outer and inner membrane of mitochondria? You call it? Between two membranes. How do you refer to it? Intermembrane. The space between the two membranes can be referred to as intermembrane space. Simple, no? So there is mitochondrial matrix, there is the inner mitochondrial membrane, and the inner mitochondrial membrane is folded. It is folded into, what do you call that? The folds of the inner mitochondrial membrane, cristae. Why are they folded? On the inner membrane you have some nails. Correct? No, when you draw the inner mitochondrial membrane, you will draw something like nails. Like somebody has from the inside hit nails on the inner mitochondrial membrane to hang clothes or to hang some things on the inside. So why all of those are there, we will see. First thing is where each of these processes is happening. Where are these processes happening? Glycolysis is happening where? Cytoplasm. Oxidative decarboxylation can happen in the cytoplasm also, happens in the mitochondrial matrix also. Krebs cycle happens in mitochondrial matrix. Electron transport chain is happening in the inner mitochondrial membrane. Simple. Alright. Why is it designed like this, we will see. Why is it designed to happen in these respective places? Glucose is produced by the cell, or is glucose coming into the cell? Is glucose produced by the cell, or is glucose entering the cell? Glucose is entering the cell. Where will it enter into? When it enters the cell, what is it entering in the cell? It is entering the cytoplasm. It is entering the cytoplasm. So in the cytoplasm, you are breaking down glucose to pyruvic acid. In the process, you are using 2 ATP, producing 4 ATPs, and also producing 2 NADPH. Correct? Some energy reactions are also happening. What energy reactions, we will see. Now this NADPH, ATP that is produced in the cytoplasm can be utilized by various chemical reactions in the cytoplasm. Wherever there is a need for ATP, ATP will be utilized in the cytoplasm also. NADPH has to be converted into what? ATP. NADPH has to be converted into ATP. Where should it be converted? Where is the mechanism for converting NADPH to ATP present? It is in the electron transport chain on the inner mitochondrial membrane. So pyruvate enters the mitochondrial matrix, becomes acetyl-CoA. Acetyl-CoA is further broken down to produce more energy compounds, 6 NADPH, 2 FADH2. 6 NADPH and 2 FADH2 is produced in the Krebs cycle for every molecule of glucose. Now, NADPH is produced more in the Krebs cycle. NADPH is produced less in the glycolysis pathway. If you had Krebs cycle in the cytoplasm, what would have been the problem? Now, because Krebs cycle is happening inside the matrix, the pH that is easier to the process which can receive which can bring about dehydrogenation and release electrons. So closer to that, you don't have to transport NADPH from outside to inside and move it here. Suppose Krebs cycle was happening in the cytoplasm. Now, because glycolysis is happening in the cytoplasm, what are you bringing inside? You are bringing pyruvate, or acetyl-CoA is coming inside. Acetyl-CoA is required outside also. Pyruvate comes in, acetyl-CoA is produced. You are only bringing in 2 NADPH inside, if required. You are only bringing in 2 NADPH inside if required. Why am I telling if required? NADPH also can be used as energy compound for various metabolic processes. We are calculating NADPH to be producing 3 ATPs. But what NADPH is produced as a part of glycolysis mostly does not even enter the mitochondria for ATP by other metabolic reactions as NADPH only. NADPH, dehydrogenase enzymes, can break down NADPH and release the energy. You understand? So, assuming that the NADPH also enters, for ATP production, maximum energy that is harnessed is in the form of NADPH and FADH2 produced during Krebs cycle. So, having the Krebs cycle happen in the mitochondrial matrix increases your chances of using that NADPH and FADH2 for ATP synthesis by passing it through the electron transport chain. Why not electron transport chain also happens in the cytoplasm? Why do we need to have it in the mitochondria? It can also happen in the cytoplasm only, no? It is a set of complexes. What is required for the flow of electrons? If electron has to flow from A to B, what is necessary? Will it just flow like that? What is required? What is required? You require a gradient. What gradient? Gradient of electronegativity. More electrons means higher electronegativity or lower electronegativity? More electrons means higher electronegativity or low electronegativity? Somebody doesn't have electrons. Who is more electronegative? I am more electronegative. So the one who receives is less electronegative. Correct? So you need to have a series of chemicals in the decreasing order of electronegativity who can receive electrons. Who can receive electrons from the one who is having electrons. So that is the electron transport chain, no? You are transporting electrons from one chemical to another chemical. When electrons move from a high energy state to a low energy state, what happens? Electron flow from A to B. Will it happen with absolute conservation of energy? Energy will be lost when electrons move from one chemical to another chemical. So in this process, the lost energy can be utilized for something. What is the lost energy utilized for in the electron transport chain? Does any of you remember? Do any of you remember? What is the lost energy of electrons as the electrons move utilized for in the electron transport chain? You can't find answers in the book. Close the book. Don't read while I am telling you some things. Alright? The earlier you learn to listen, the better it is. Second time. I remember. Alright? Trust in the guy. Alright. So what is it? What is the energy of the electron that is lost in the transfer used for? Pumping protons, pumping hydrogen ions. Alright, remember this much, enough. So we need to deal with gradient utilization and gradient creation. Gradient utilization and gradient creation requires compartmentalization. So from the inside of the mitochondrial matrix, you are pushing protons outside of the mitochondrial matrix. If you push protons outside of the mitochondrial matrix, where will the protons go? Will it go out of mitochondria? Where will the protons go? Into the intermembrane space. When you push protons from the mitochondrial matrix outside of the matrix, it goes to the intermembrane space. Alright, if you remember, There are 5 complexes in electron transport chain. 1, 2, 3, 4 and the 5th one is called ATP synthase. ATP synthase is the only complex among these which will allow protons to come back. It is like a generator. You have seen hydroelectric power stations. How do they operate? You have a rotor. You have a rotor. As the water falls on the rotor blades, the rotor will rotate and that will lead to production of electricity by the generator. ATP synthase is like that. What is the water that has to flow? It is the hydrogen ions or protons. When protons flow through the ATP synthase complex, imagine it to be like this. There is ATP, ADP, phosphate. The ATP synthase complex has 2 arms to catch hold of these 2. It's an enzyme too. ATP synthase is also an enzyme. It has 2 arms. When hydrogen ion flows through this, these 2 arms come closer to each other. You understand? And there is energy of electrons also. There is energy there. And when ADP and phosphate comes closer to each other, this is leading to the formation of ATP. Then one more proton flows in, brings ADP and phosphate, ATP synthesis. Proton flows in, brings ADP and phosphate, ATP synthesis. ATP synthase can do this only by allowing protons to flow inside, outside to inside. When protons flows from outside to inside, ATP synthase can bring ADP and phosphate together to produce ATP. Now for protons to flow from outside to inside, what do you need to create? You need to create a gradient for protons across the inner mitochondrial membrane. Correct? So the gradient, the number of protons, the concentration of protons in the intermembrane space should be more than the concentration of protons in the mitochondrial matrix. That will create a gradient which will make the protons flow inwards. Are you understanding? Is everybody understanding or not? Who is not understanding? Raise your hands. I am just giving you an overview. We will go into details. If anybody has not understood, raise your hands. Everybody understood? Okay, good. So 2 gradients we are working with. 3 things are happening. 3 things are happening in electron transport chain. First, you have to release electrons. Somebody should break down something, ionize it and release electrons. Now these electrons will start flowing from one compound to another compound to another compound, following the electronegativity gradient. Electron flow should happen. As the electron flows, there is energy released. That energy that is released is used for pumping protons outside. The energy released is used for pumping protons outside of the matrix into the intermembrane space. Now when there is a gradient, the protons want to come back. The protons want to come back into the mitochondrial matrix to bring about an equilibrium. They cannot come back through anything else, but they can come back only through ATP synthase complex. They can come back only through ATP synthase complex. Those nails that you see, no? Those are the ATP synthase complexes. So they can come, the protons can come back only through the ATP synthase complex. Now when that happens, ATP synthesis happens. There is another problem. As long as electrons keep flowing, see, electron flow is being created, electron flow is happening to serve 2 purposes. One, to push protons outside of the, to liberate energy, which will push protons from the matrix to the intermembrane space. Electron flow can continue to happen as long as there is gradient. Alright? Let us say there are 6 people. Okay, we will do this. Come here. You also come here. Come. You also come. Alright? Alright. No, I don't need 5 people. 4 people. You sit, ma. Stand properly. Stand at a distance. Now each of the complexes. Everybody can hold a bunch of electrons, let us say. Electron flow will start from there. So among these four, let us say 1, 2, 3, 4. Who is the most electronegative? He is the most electronegative. I will do one more thing. You stand a little backwards. All of you come forward. All right. I want somebody else also. Okay, come, stand here. Stand there in the front. There. Now, electrons are obtained here. Pass it on to her. Give it to her. Just pass on the electrons. You give here. You give here. Okay. He is also producing electrons. You give the electrons to her. Give. Give. Now each one can hold a bunch of electrons. What did you observe so far? He is 1, 2, 3, 4. What is your observation so far, you tell me? Do it again. Okay. What is your observation? One intermediate is there. So 1 and 2 are not giving electrons to each other. 1 is not giving electrons to 2. Electrons that are produced at 1 through an intermediary is coming to 3. Electrons produced by 2 through an intermediary is coming to 3, then to 4. All right. Now, spread out. Spread out. Come forward. Come forward. You stand there only. You stand back. Okay. Spread out. I want 4 more. I want 3 more people. 3. All right. You all stand backwards. You go there. There. No. Go there. One of you go there. You stand there. You come here. All right. These 3 people whom I have brought now are protons. That is the matrix. This is intermembrane space. That is the matrix. This is intermembrane space. Now, 1, pass on the electron. Come forward. Proton, come forward. You are not proton. You are the intermediary. Stand there. Proton, come inside. Come into the intermembrane space. 2, pass on the electron. Nothing happens. Pass on the electron to 3. Come. Pass on the electron to 4. You come forward. Now what happens? I am just talking about passing on the electrons. You should imagine electrons flowing through the complex. When electrons flow through complex 1, what will happen? One proton moves out into the intermembrane space. When electrons flow through complex 2, what happens? Nothing happens. When electrons flow through complex 3, what happens? One proton is coming into the intermembrane space. Same with 4. Now, 1, 3 and 4 are capable of pumping out protons from matrix to the intermembrane space. 2 is not capable of doing that. All right. 2 is not capable of doing that. So if electron donor gives electrons to 1, there is somebody who should give electron, no? If the electron donor is giving electrons to 1, that electron flowing through the entire complex will lead to the expulsion of how many protons? 3 protons. And who is the terminal electron recipient? Who will take electrons from him? If somebody does not take electrons from him, he will not be able to take the electrons from him. So next step, this fellow will be holding electrons and he will not be able to receive electrons because electronegativity will increase. With you are holding on to electrons, your electronegativity increases. And you can't receive another electron because you don't have the gradient for it. Then it will stop, shut down. The entire electron transport chain will stop. If electron transport chain does not happen continuously, what will happen? ATP production? ATP production will not happen. If there is no ATP, metabolism will not happen. If there is no metabolism, that is death. Absence of metabolism is death. Biologically, absence of metabolism is death. Continuous metabolism is life. Existence of life is proved in the presence of metabolic reactions happening in a coordinated manner. If the reactions stop, functions stop, then there is death of the body. So, somebody should receive electrons from him. If there is no electron recipient, terminal electron recipient, we will die. Who is the terminal electron recipient? Who receives the electron from complex 4 to ensure that this process is continuously happening? Absence of what will kill us instantly within a few minutes? Oxygen. Why do we die if we don't have oxygen? Oxygen takes electrons from him. Is oxygen highly electronegative or low? It is low in electronegativity. Oxygen's electronegativity is low. Oxygen's electronegativity is low. So it can receive electrons from him. So low and high is relative. So this person's electronegativity is higher than the electronegativity of oxygen. Similarly, higher and higher and higher as we go to the first complex that receives the electrons. Oxygen is the terminal electron acceptor. The terminal electron acceptor is oxygen. I am oxygen now. Give. Then you collect it from him. Now again, I am receiving electrons. What should I do? I receive electrons. I am there. I will combine with proton and I become water, metabolic water. At this complex, at complex number 4, electron is being finally delivered to oxygen, who is the terminal electron acceptor. Oxygen, proton and the electron that is received combines to form water, metabolic water. This is electron transport chain. You don't have to know any names. That's what I tell you. There is a story before facts. There is a story before names. You should get the story first and then link the names. If you try obsessing about names, you will miss the story. You should not miss the story. You get this now? Go back, all the complexes go back. Thank you very much. Did you get a picture of electron transport chain? Glycolysis and Krebs cycle is a series of chemical reactions. Easy to understand, no problem. Fundamental principles, I'll tell you. But electron transport chain is complex. Complex because there are too many things. Like this arrows, too many arrows. Somebody is losing electrons, somebody is gaining electrons, somebody is, or redox, series of redox reactions. Somebody is getting reduced, somebody is getting oxidized. So many weird names and different ways of putting it. Those arrows will make you feel all right. Even there, it is simple. You have a series of chemicals. One chemical gives electron, it becomes oxidized. The other one receives electrons, it gets reduced. Goes on like that. It's the same series. The same one which gets reduced, which received electron, gives electrons to the next one. So it's the same series that we will see later, not today. Alright, today I want you to get the story. Is this clear? Electron transport chain, you understood? It is the same mechanism that happens in chloroplast also. But there the gradient is reverse. Here, matrix to intermembrane space. In chloroplast, it is a reverse gradient. It is from outside to inside the gradient is created. Gradient is created for inside to outside. So protons are pumped inwards. We will see that when we go there. Alright. Now, with this background, I will take you through the notes that I have prepared for you for this question, for the question. That will help you understand these processes completely. So what did we understand now? Where are these three processes happening? Glycolysis happens in cytoplasm, Krebs cycle happens in mitochondrial matrix, electron transport chain is happening on the inner mitochondrial membrane. Inner mitochondrial membrane. So the flow of electrons are happening in the where? In the membrane, in the inner mitochondrial membrane. Protons are pumped from where to where? Protons are pumped from where to where during electron transport chain? From matrix to intermembrane space. Gradient is from where to where? The proton gradient is from where to where? From the intermembrane space to the mitochondrial matrix. Gradient is from the intermembrane space to the matrix. How many complexes are there? How many complexes are there? 5 complexes. The 5th one we also call as ATP synthase complex. Each one has a different name. So 1, 2, 3, 4 and 5. 5th one is the ATP synthase complex. Which complexes? Now I am asking you something which you have to answer on the basis of analysis of what we have discussed, because I have not told that directly. There are 4 complexes embedded on the inner mitochondrial membrane. Which of these 4 complexes do you think are through the membrane, that is extending throughout the membrane, having its exposure on both sides of the membrane? Which complexes are extending throughout the membrane, across the membrane? Of 1, 2, 3, 4, which complexes are extending throughout, across the thickness of the membrane? 2. Okay. Why? Why do you say so? If this is the thickness of the membrane, inner mitochondrial membrane, which complexes are throughout the entire thickness of the membrane? That's from this side to this side it is extending. ATP synthase, yes, because it has to let the protons flow through it. Then apply the same logic. Apply the same logic. Complex 4. Why? Electron? You have to be loud. See, I have told you, I have been shouting for a long period of time, over 2 decades. So as a consequence of that, I have become a bit deaf. So be louder, shout out your answers. What is the logic behind you telling ATP synthase is extending throughout the membrane? Hydrogen ion has to pass through it from the inner intermembrane space to the matrix. Okay. Which other complexes are extending throughout? Extrapolate the same logic. Think about it in a different way. What will the complex be able to do if it is throughout the membrane? If it is across the membrane, what is it that the complex can do? Or what it cannot do if it is not across the thickness of the membrane? See, you can never be prepared for questions. You have to be prepared for logic. It is the same concept, but you have to apply logic. Answer is already told. He has already told the answer. If I say except complex 2, all the other complexes are extending through the membrane. Justify. Except complex 2, all the other complexes are extending throughout, are extending through the thickness of the inner mitochondrial membrane. How do you justify that? Justify that with some explanation. Protons are being pumped from the from the mitochondrial matrix to the intermembrane space. In which complexes? In which complexes are protons being pumped from matrix to the intermembrane space? 1, 3, 4. Coming back is through ATP synthase. 2 is not extending throughout the membrane. So it cannot allow proton to flow from inside to outside. Alright? So think. When you start giving explanations by logic to things which are not outrightly taught to you, that is when the actual preparation begins. Alright? You should be able to apply some logic and arrive at some sensible logical explanation to a situation. The explanation might be wrong, but it should be based on some logic that generates in your head. That is when your preparation for success in NEET or any competitive exam starts. As long as your preparation is, as long as your approach is limited to remembering things and trying to recall, preparation has not yet begun. Alright? So you have to break the pattern of trying to remember things. Don't try to remember things. Try to generate logic. Generate logic, it might be wrong, but generate logic. Analysis. Alright, those are all terms which are used. But unless we go through a process of stimulating those abilities in our mind, we will not be able to do it spontaneously. Oh. Alright, so we'll go through the notes now. We'll go through the notes. Notes is designed to help you ask some questions. How do we make this go away? So the three boxes in this diagram represent the three major biosynthetic pathways of aerobic respiration. Arrows represent net reactants or products. We analyzed this diagram yesterday. We will do it again. Glucose enters the pathway. Pathway A is glycolysis. Pathway B is Krebs cycle. Pathway C is electron transport chain. Pathway C is electron transport chain. Is the mic working? Okay. So 4, 8 and 12. What is represented by 4, 8 and 12 is the question. Pathway A is like, all of you have your papers also, no? So for cross-referencing with the diagram, you just look into the paper that you have. Pathway A is glycolysis, pathway B is Krebs cycle, also known as citric acid cycle. Why citric acid cycle? The first stable product formed during Krebs cycle is citric acid. So that is why it is also referred to as citric acid cycle. Pathway C is electron transport chain. What do the arrows represent? Arrow 1, glucose to pathway A. Already glucose is entering. The diagram is not good. The diagram is not good. It is mentioning glucose and it is also mentioning arrow 1. What should we consider arrow 1 to be then? We should consider it to be ATP, because in the first reaction of glycolysis, ATP is used in the conversion of glucose to glucose 6 phosphate. So we can assume that to be ATP also. Arrow 2, the products of glycolysis, which is pyruvate, moves to the Krebs cycle. Arrow 3 likely represents NADPH, which enters electron transport chain to further transfer energy for the production of ATP. You look into the diagram that you have. Arrow 3 should be NADPH, because you see from pathway A, something is moving into pathway C. There are three things that are produced in glycolysis. One is pyruvate, one is pyruvate which goes into Krebs cycle. So 2 has to be pyruvate. ATP is produced directly. 4 has to be ATP. NADPH is produced. NADPH goes into electron transport chain. So if you see 3 is going into pathway C, electron transport chain. So 3 has to be NADPH. Similarly, if you look at pathway B, from Krebs cycle what is being produced? You have carbon dioxide as a byproduct. It produces carbon dioxide. You have NADPH, you have FADH2, and also GTP. 4 things are produced. So you have 3 being represented here, 6, 7, 8. 9, 10 also is being represented. 9, 10 outside of pathway B. It is not coming from pathway B. 9, 10 is coming from somewhere else. Correct? So, what is arrow 5? Likely represents acetyl-CoA which enters the Krebs cycle. You see this? Acetyl-CoA can enter from other places also, not necessarily from pyruvate. Acetyl-CoA is a byproduct in protein metabolism, fat metabolism as well. So, acetyl-CoA can directly enter Krebs cycle. 6, 7, 8. 6 represents NADH or FADH2 which carry electrons to the electron transport chain. 7 represents carbon dioxide released during Krebs cycle. 8 represents ATP, which is ATP generated in the Krebs cycle. 9 represents electron transfer to the electron transport chain. 10 represents oxygen entering the electron transport chain. Alright, so we saw that in the diagram. 9 and 10 coming from outside to the electron transport chain. So if you consider one of this, 9, let us say 9 is electron donors coming into the electron transport chain. NADPH and FADH2 are not produced only by glycolysis. They are produced in other pathways also. So they also can enter into the electron transport chain directly. Alright. So they are electron donors coming into the electron transport chain. And also for electron transport chain, we require oxygen. Oxygen is also entering pathway C. 9 and 10. 9 can be considered as high energy compounds or electron donors entering the electron transport chain, which were not produced in the Krebs cycle or glycolysis. So basically NADPH and FADH2 produced elsewhere. 10 is oxygen. 11 and 12 can be water and ATP. That's all, no? Water is produced as a byproduct of, as the end product of electron transport chain. ATP is the end product of energy transformation in the electron transport chain. So these are the 12 things that are represented in the diagram. Do you understand all of this? You should not, there is no point of remembering. There is no need to remember. Glycolysis, glucose enters it. ATP is also entering the first reaction. So we can consider 1 to be ATP. Pyruvate getting into Krebs cycle after getting converted into acetyl-CoA. Acetyl-CoA is produced in other pathways also. So acetyl-CoA can enter Krebs cycle directly. So all acetyl-CoA that enters Krebs cycle is not a byproduct of glycolysis. It is not necessarily coming from glycolysis. Acetyl-CoA can come from other pathways also. So what can enter Krebs cycle is acetyl-CoA. Pyruvate gets converted to acetyl-CoA and enters Krebs cycle. Acetyl-CoA can enter Krebs cycle directly also. energy, two different energies produced by glycolysis, ATP, NADPH goes into ETC. Same with Krebs cycle, carbon dioxide is produced, NADPH, FADH2 is produced. This is again wrong, because one of the arrows, this 7 or 6 should have been connected to this. Correct, no? Only then we can consider that to be NADPH or FADH2. See the error in this? Do you see the mistake in this? 6, 7, 8. None of these are going into pathway C. Two can be explained. One of the 3 is carbon dioxide, gets out. One of the 3 is GTP, doesn't get into electron transport chain. But one of the 3 is certainly NADPH and FADH2. It should enter electron transport chain. But this diagram is not representing that. So, error. 9 and 10, fine, we can explain that, because high energy compounds, electron donors, can come from outside of this sequence also. So, one of that is that. And one is oxygen, which will contribute to, which will be the final electron acceptor, terminal electron acceptor. So, that is also explainable. These two are also explained properly. So, mistake in this pathway is here 2 should have been shown as getting converted into something else that enters the Krebs cycle, and that something else would be what? What is that something else 2 should be converted to? Acetyl CoA. Acetyl CoA. Alright. Clear? Now, how many carbons? The removal of how many carbons from glucose is contributed by glycolysis. How many carbons are removed in glycolysis? How many carbons are removed in glycolysis? 0. How many carbons are removed in Krebs cycle? Per molecule of glucose? How many carbons are removed as carbon dioxide in Krebs cycle per molecule of glucose? 4. Where are the other 2 carbons removed? In oxidative decarboxylation, pyruvate to acetyl CoA. This is also something that can come as question. All right, number of carbons. Where is carbon entering? Where is carbon exiting? Important. In photosynthesis, you should know the reactions where carbon enters the system. It comes as carbon dioxide, gets incorporated. In glucose metabolism, you should remember which are the reactions where carbon exits the system as carbon dioxide. All right, important. You should also remember in this number of carbons, which compound is how many carbons, which chemical is how many carbon compound. All right, so we will see that. All right, we'll go down further. Arrows, analysis of the diagram is done. We analyzed all the arrows there. We also identified what are the errors in the diagram. All right, when you do analysis, you will get all of this. Pathways in aerobic respiration, glycolysis is pathway A, produces 2 molecules of NADH, produces 2 ATP, which is net gain, and 2 molecules of pyruvate. Simple. It's all there in one thing, but this is perspective notes. This is perspective based notes in the same question analysis. What is being produced? That's all. That is what we will look at in this part. Krebs cycle is pathway B. See, when 2 ATP is the net gain, if the question is how many ATPs are produced, how many ATPs are produced in glycolysis, energy dynamics of glucose metabolism. Glycolysis is the process of breaking down 1 molecule of glucose into 2 molecules of pyruvate. Simple. ATP production, gross ATP produced 4 ATP, net ATP produced 2 ATP. Gross is what you can calculate before taking into account expenditure. Net is what you calculate after taking into account expenditure. In simple business financial terms. NADH production, total NADH produced 2 NADH from the oxidation of glyceraldehyde 3-phosphate to 1,3-glyceraldehyde bisphosphate. Enzyme is G3P dehydrogenase. Very important enzyme. We will come to that. FADH2 production, total FADH2 produced in glycolysis, 0. No FADH2 is produced. So summary for glycolysis, net ATP, 2 ATP, NADH, 2 NADH, FADH2, 0 FADH2. This is the energy dynamics. You have to look at everything in one perspective. So all three we looked at in terms of matter transformation. What is being converted to what? Now we are looking at it in terms of, in each of the processes, energy dynamics. Right? Krebs cycle, citric acid cycle, occurs twice for each molecule of glucose. That means once for each pyruvate. ATP produced, 2 ATP. 1 ATP per cycle. It is actually GTP that is produced. We can consider GTP to be equal to ATP. So GTP is formed and is often considered equivalent to ATP. NADH production, total NADH produced, 6 NADH, 3 per cycle. Okay. FADH2, 2 FADH2, 1 per cycle. Summary for Krebs cycle, how many ATPs? 2 ATPs, 6 NADH, 2 FADH2. Overall summary. Krebs cycle, 4 ATPs, 2 from glycolysis, 2 from Krebs cycle. 8 NADH, 2 from glycolysis, 6 from Krebs cycle, 2 FADH2, all from Krebs cycle. So in terms of ATP, these NADH and FADH2 molecules are then used in the electron transport chain to produce additional ATP. For considering the energy produced in ATP equivalent, how many molecules of energy equivalent to ATPs are produced? We have to consider 1 NADH to be equal to how many ATPs? 3 ATPs. 1 FADH2 is equal to 2 ATPs. So what is the total ATP production per molecule of glucose? 4 plus 8 into 3, 24, plus 2 into 2, 4. How much? 32. But what is the total number of ATPs that we get from one molecule of glucose? 38. Where are the 6 ATPs missing? Where are the 6 ATPs? Oxidative decarboxylation gives you 1 NADPH per pyruvate to acetyl-CoA conversion. So it happens twice per molecule of glucose. So 2 NADPH is coming from oxidative decarboxylation. Add that into 3, gives you 6 ATPs, totally 38 ATPs. So NADPH, total NADPH produced in the metabolism of glucose is 10. 2 in glycolysis, 2 in oxidative decarboxylation, 6 in Krebs cycle. 2 FADH2, both in Krebs cycle, 4 ATPs, 2 in glycolysis, 2 in Krebs cycle. Total 38. Alright. Is it clear so far? Matter transformation, energy transformation, and basic understanding of electron transport chain. Clear so far? Yes or no? Okay. Any questions so far? Anything that you want me to repeat at this stage? Glycolysis, oxidative decarboxylation, Krebs cycle, ATP, NADPH, FADH2. Alright, we can make another small table for this. I am very fond of tables. Helps us summarize things properly and compare things efficiently. How many ATPs in glycolysis? How many ATPs in glycolysis? How many ATPs in glycolysis? Net. Talk about net. 2. How many NADPH in glycolysis? And when we are talking about all this, we will talk about it for one molecule of glucose, not one cycle. How many molecules of NADH in glycolysis? 2. How many molecules of FADH2? 0. In decarboxylation, how many molecules of ATP? How many molecules of ATP are produced in oxidative decarboxylation? 0. NADH? 2. FADH2? 0. ATP in Krebs cycle? 2. 1 GTP per cycle. So Krebs cycle happens twice for one molecule of glucose. How many NADPH in Krebs cycle? 6 NADPH. How many FADH2? 2. Add total ATPs 4. Total NADPH 10. Total FADH2 2. You can also add ATP equivalents. ATP equivalent. So in terms of ATP, how much does it mean? 4 ATPs is equal to how many ATPs in terms of ATP? 4 ATPs is equal to how many ATPs? 4. 10 NADPH is equal to, you can mention into 3 on the top. So it will be 30 into 2 on the top. This will be 4. So total ATP per glucose is 38. Lot of questions come on this. Simple questions, outrightly answerable questions. We will not be able to answer because clarity of understanding is not there. All the tables that I give are all tables that I create. You will not find those in these books. Alright. So you will find these tables only in my book when I come out with the book. I'm promoting it so that you can tell your juniors to buy it. Alright. Okay. Done? Understood? Calculation part? I'll erase this. We can do one more here. Carbon dioxide released. Glycolysis, oxidative decarboxylation, Krebs cycle. How many carbon dioxide is released in glycolysis? 0. In oxidative decarboxylation? 2. Krebs cycle? 4. This is also something we need to know. So if how many reactions then? We have to remember how many reactions for understanding where carbon dioxide is being released. Oxidative decarboxylation happens twice per molecule of glucose. So it is one reaction. Krebs cycle happens twice for one molecule of glucose. So if 4 carbon is released, that means you are having the reaction 2 times. So only 3 reactions we have to remember. We have to remember 3 reactions clearly where carbon is being removed. That means the substrate for that reaction will be a compound which is 1 carbon higher than the product. So if the substrate is a 4 carbon compound or a 5 carbon compound, the product will be a 4 carbon compound. That you have to remember. Questions can be so many compounds that are present in these cycles, and asking you to pick the odd one. The odd one can be based on the number of carbons that each of these compounds have. You can be given reactions and you can be asked to pick the odd one. The odd one can be a reaction where carbon is being released. You can be asked questions where reactions are given, reactions which are producing NADPH, reactions which are producing FADH2. The one which is producing FADH2 can be the odd one when the others are NADH producing reactions. You can have reactions where NADPH and FADH2 producing reactions are given, and one reaction which is producing ATP. The ATP producing reaction becomes the odd one. You understand? You can be given enzymes and you are asked to pick the odd one. So enzymes can be either all the enzymes related to a particular sequence of reactions, particular process or pathway. So there can be enzymes of glycolysis with one enzyme of Krebs cycle. Enzyme of Krebs cycle becomes the odd one. You can be given a set of enzymes based on their functions. You can have multiple kinases with one dehydrogenase. Dehydrogenase becomes the odd one. Kinases are all associated with phosphorylation. Dehydrogenase is associated with proton transfer. Alright. So questions can be formed with the same topic, with the same concept. You can create 100 different questions. You understand? So the basis for answering all that question is a clear understanding of the concept. Okay? Doing too many questions and trying to remember answers. Big problem that I have with the training methodology that most of you are exposed to, and most of it goes on, is trying to make the students go through a lot of questions and remember the answers. If you have solved many questions like this, when a question of the same kind comes, you will be able to tell the answer. And even training students on identifying key points and arriving at the answer without even going through the process of solving it. If this are present in a question, the answer should be this. Shortcuts. The most effective shortcut would be that which will help you arrive at answers logically. Logic will take you to an answer very quickly. Yes, sir. What?
For the NADH, travel from the glucose to glycolysis to the electron transport chain.
It will lead to ATP production. NADPH is an electron donor. Where it is coming from doesn't matter. Because we are studying glucose metabolism, we are looking at NADPH being produced here. NADPH can come from anywhere. Electron transport chain requires electron donor, electron accept, that's all, and a proton gradient. If these three things are, the electron transport chain will run. Alright. Where the NADPH comes from doesn't matter. Okay. If it is clear so far, let us move ahead. Shall we? See, glycolysis is a series of 10 reactions. You would have seen glycolysis being represented like this. Glucose, glucose 6-phosphate, ATP, ADP, then hexokinase, like this. Goes on for every reaction. Correct? Not easy to understand. For any reaction, there is something that enters the reaction, something that exits the reaction. If you look at this, This is entering the reaction. Glucose is entering the reaction. Glucose 6 phosphate is exiting the reaction. This is the beginning of the reaction. This is the end of the reaction. ATP is entering the reaction. ADP is being produced. But this is related to energy. So I can say energy is utilized. Energy is used here. When ATP is being broken down, energy is used. When ATP is produced, energy is produced. And there is an enzyme. So I have given the reactions of glycolysis and Krebs cycle also in a table format. The table comprises this. Serial number. I want you to remember the reaction numbers. Reaction numbers I want you to know. What enters the reaction, what exits the reaction, what is the enzyme, is it reversible or not? Very, very important. Questions will come on this. Energy used, if energy is used or energy is produced, net energy in terms of ATP equivalent. If you see this, if energy is used, net energy will be in negative. Clear? Are you understanding the design of the table? Now we will go through it reaction by reaction. The first reaction is glucose getting converted into glucose 6 phosphate. Meaning what? A phosphate is being attached to glucose at carbon 6 position. That's all, no? Glucose 6 phosphate means it is being attached to glucose at position number 6. Now that means you need energy to create the bond between glucose and phosphate. Energy should come from somewhere. It is an energy consuming reaction. And you need phosphate. You need phosphate group coming from somewhere for the reaction. You need energy to link the phosphate with glucose. That we need to understand when we look at the substrate and product. Enzyme is there, let it be. Energy is coming from ATP. Not only energy, phosphate is also coming from ATP. The advantage of using ATP for this reaction is it will provide the energy for the reaction, and also when ATP is broken down, phosphate group is released, the same phosphate group is attached to the glucose. So glucose becomes glucose 6 phosphate. The reaction is irreversible. You should remember, irreversible reactions are the ones which will ensure that the pathway goes on in a direction. And I want you to remember I want you to think about one more thing about the enzyme. Every enzyme is capable of carrying out reactions in both directions. Now hexokinase, whenever kinase is the enzyme, kinases will bring about phosphorylation. It will act on the phosphate bonds. Alright, kinases will bring about phosphorylation. You have hexokinase, phosphofructokinase. You have fructose 6 phosphate being converted into fructose 1,6 bisphosphate. It is a phosphorylated fructose on which the enzyme is acting. So the enzyme is phosphofructo. Its action is to phosphorylate at position 1. So the nature of the enzyme is kinase. That is why the enzyme is named as phosphofructokinase. It is acting on phosphorylated fructose and attaching a phosphate group to it. By virtue of attaching phosphate group, it is referred to as kinase. And because it is acting on phosphorylated fructose, the enzyme name is phosphofructokinase. Name, understand what the name means, a lot of things become automatically clear. And you can name it as you go forward. Alright, this is also irreversible. And if you notice, go down further. I'll come back to every reaction. We'll look at the next irreversible reaction. See the reaction number 10. Phosphoenol pyruvate is becoming pyruvate. Here if you see, in the earlier two kinase reactions, phosphate got added to the substrate. Kinase added phosphate to the substrate. In reaction number 1, glucose got a phosphate group and became glucose 6-phosphate. In the next phosphorylation reaction, fructose 6-phosphate got a phosphate group and became fructose 1,6-bisphosphate. Both the reactions were mediated by kinase, hexokinase or glucokinase, and the second enzyme was phosphofructokinase. We have one more kinase here, pyruvate kinase. What is it doing? It is removing phosphate from phosphoenol pyruvate and making it pyruvate. I told you every enzyme can do reactions in both ways, and this is also irreversible reaction. Glycolysis has 3 irreversible reactions. What is the speciality of all the 3 reactions? All the 3 are kinase reactions. All the 3 are kinase reactions. That means all the 3 are reactions which are ATP dependent, phosphorylation dependent. All right. There is one more kinase here, phosphoglycerate kinase. Phosphoglycerate kinase. It is a reversible reaction. You see, 1,3-bisphosphoglycerate is being converted to 3-phosphoglycerate. Here also, phosphate is being removed. So when phosphate is being removed, the energy of breaking it down is used to take this phosphate and give it to ADP. You understand? Phosphate is being dealt with ATP only there. When the substrate needs to be given phosphate, you take the phosphate from ATP, use the energy of breaking ATP to link this phosphate to the substrate. If you are breaking the phosphate from the substrate, use the energy to link this phosphate to ADP to make ATP. Are you understanding? Break ATP, use the energy, attach the phosphate to the substrate. Break the substrate, use the energy, take that phosphate and give it to ADP, you make ATP. Both these kind of reactions are mediated by kinases. So how many kinase reactions are there in this? 4 kinase reactions. 2 kinase reactions are breaking ATP. 2 kinase reactions are making ATP. You understand? All the 4 are kinases. Of the 4 kinase reactions, 3 are irreversible reactions. Very important. So these enzymes which are mediating the irreversible reactions are important enzymes. Alright. So forget about the names. I will tell you the substrate and the product. You tell me the enzyme name. Glucose, don't look at the board. Glucose gets converted to glucose 6-phosphate enzyme. Hexokinase or glucokinase. Fructose 6-phosphate is converted to fructose 1,6-bisphosphate. Phosphofructokinase. Phosphofructokinase. 1,3-bisphosphoglycerate. 1,3-bisphosphoglycerate is converted to 3-phosphoglycerate. Enzyme is bisphosphoglycerate. That is phosphorylated glycerol. Phosphorylated glycerol is the substrate that is being acted upon. So the enzyme is kinase. The enzyme should be phosphoglycerokinase. That's all, no? What is the enzyme there? Phosphoglycerate kinase. Okay. Phosphoglycerokinase or phosphoglycerate kinase. Which is the 4th kinase reaction? Phosphoenol pyruvate is converted to pyruvate. So the enzyme is pyruvate kinase. Why are we calling it pyruvate kinase? Why are we not calling it phosphopyruvate kinase? Kinase doesn't mean attaching phosphate to the substrate. You can name the enzyme either way, the substrate or the product. Use whatever is convenient. Phosphopyruvate kinase is more complex than pyruvate kinase. So pyruvate kinase is simple, name it that. Understand? So you know 4 reactions already. There are 4 kinase reactions. Alright, now we will look at the reactions one by one. All the energy linked reactions, ATP associated reactions we have seen. We will see the rest of the reactions also. Clear so far? You cannot just, glucose is eventually broken down into pyruvate too. Done. But if you join 2 pyruvate, you will not get glucose. Chemically, glucose is not pyruvate plus pyruvate. You understand this? Chemically, glucose is not pyruvate plus pyruvate. So glucose is a 6-carbon compound. You cut glucose at 3 carbons. You have 1 pyruvate, another pyruvate. No, that's not it. The structure of pyruvate is not similar to glucose. To make sure 3 carbons of glucose is altered and brought into 3 carbons of pyruvate, the other 3 carbons of glucose is brought into the 3 carbons of pyruvate, you will have to take it through steps which will change the structure sequentially so that by the end of it you have pyruvate. Alright? That is why we have so many steps. A metabolic pathway, you have the beginning of the pathway and you have the end of the pathway. The end and beginning are not necessarily related. So transforming the beginning to make it arrive at the end needs transitioning of structure. Structure should keep transitioning, it should keep changing. Stage by stage, step by step, the structure keeps changing. That is why these things are there. So many reactions are there. Alright? Is it clear? We'll go one by one. See, don't sit without understanding. If you are not understanding something, stop me, ask me. If you are not stopping me or asking me to repeat or explain it again, I will assume that you are understanding. So the onus is on you to ensure that you are understanding. Enzymes which are mediating irreversible reactions. All other enzymes are mediating reversible reactions in glycolysis. So reaction number 1, enzyme, hexokinase. Reaction number 2, enzyme, isomerase. 3 is fructose 6-phosphate is now becoming 1,6, fructose 1,6 bisphosphate. Enzyme has to be some kinase. The enzyme has to be kinase. What kinase? Phosphofructokinase. Energy used because the product has one more phosphate attached to it. Attaching something to something requires energy, and because it is phosphate that is being attached, the phosphate is coming from ATP. Energy used is 1 ATP. Energy produced is 0 ATP. Net is -1. Alright. So we are done with 3 reactions. Reaction number 4. You see this? Fructose 1,6 bisphosphate is a how many carbon compound? It's a 6 carbon compound. Now in reaction number 4, we initiate the process of breaking down the 6 carbon into 3 carbons. Alright, here fructose 1,6 bisphosphate is broken down into 2 3 carbon compounds, glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. Both are aldehydes. Glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. Both are aldehydes. Enzyme which is breaking down a substrate to produce aldehydes is the enzyme called aldolase. Is it, remember? Aldolase. Now glyceraldehyde 3-phosphate is the one which will go through further sequencing, further transformation to become pyruvate. Glyceraldehyde 3-phosphate will go through further steps to become pyruvate. But along with glyceraldehyde 3-phosphate, we have one more compound, 3-carbon compound. What is that? Dihydroxy acetone phosphate. The next reaction, reaction number 5, is converting dihydroxy acetone phosphate into glyceraldehyde 3-phosphate. DHAP, dihydroxy acetone phosphate, and G3P, glyceraldehyde 3-phosphate, both are isomers. So what should the enzyme be? It should be some isomerase. What isomerase? Glyceraldehyde and dihydroxy acetone phosphate. Both are 3-carbon compounds. 3-carbon compounds can be called trioses. They are not simple trioses, they are phosphorylated trioses. So the enzyme is triose phosphate isomerase. That's all. Naming. Triose phosphate isomerase. What does it do? It will convert dihydroxyacetone phosphate into glyceraldehyde 3-phosphate. DHAP can go into other pathways also. So if there is a demand for lipid synthesis, DHAP can go into that. So G3P will be converted to DHAP in that requirement. The reaction is reversible. The reaction is reversible. All right. Next. Glyceraldehyde 3-phosphate is getting converted to 1,3-bisphosphoglycerate. Glyceraldehyde 3-phosphate is getting converted to 1,3-bisphosphoglycerate. Now, pay attention here. Glyceraldehyde 3-phosphate has how many phosphate groups? It has how many phosphate group? One phosphate group. 1,3-BPG, 1,3-bisphosphoglycerate has how many phosphate groups? Two phosphate groups. So one phosphate group is being attached. Where is the phosphate coming from? Is ATP being used here? ATP is not used in this reaction. Something has to be broken. Something has to be broken to release energy. And that energy has to be used to attach this phosphate group. But what is being broken is not ATP. So, a hydrogen is broken off from 3-phosphoglyceraldehyde 3-phosphate. Hydrogen is broken off from glyceraldehyde 3-phosphate. Hydrogen is given to NADP to make NADPH. Phosphate group, inorganic phosphate group is taken and attached to glyceraldehyde 3-phosphate. So the energy released by breaking down hydrogen is used for attaching two things to two different substances. NADP is being given hydrogen, glyceraldehyde 3-phosphate is being given phosphate. So as a product we get 1,3-bisphosphoglycerate, and as a energy compound we get NADPH. Very important reaction. This is a reaction where we are producing NADPH. This reaction could have been mediated by kinase also. If you had utilized kinase, this reaction would have been an energy consuming reaction. Because you are utilizing dehydrogenase, this reaction is an energy producing reaction. You understand it? See, evolution has improvised the mechanisms. Kinase would have done the job of converting glyceraldehyde 3-phosphate into 1,3-bisphosphoglycerate. But that would have used energy, because kinase will use phosphate from ATP, will break down ATP and attach phosphate here and use the energy for attaching phosphate. What did evolution do? What did nature do? It broke down hydrogen from glyceraldehyde 3-phosphate using an enzyme dehydrogenase. Energy is released. You break any bond, energy is released. and that energy is used for doing two jobs. One job, give phosphate to 1,3-bisphosphoglycerate. Another job, to give that hydrogen to NADP and produce NADPH. Now NADPH is equivalent to 3 ATPs. NADPH is equal to 3 ATPs. So using kinase, what would have been an energy consuming reaction became an energy producing reaction by just changing the nature of enzyme that you are using for doing this. Alright? Improvisation. Evolution is improvisation. Evolution is better process, developing better processes to achieve the outcomes. So evolution is not perfection. Evolution is a process of continuous improvement in effectiveness. With doing things more and more repeatedly, you improvise and you become more effective. What you are going through is also a process of evolution. Doing the same things again and again and again and again, improvising your efficiency, improvising your work, you will become more and more effective in doing this. And hopefully, most certainly, if you follow the process, by the end of this phase, all of you would be capable of spontaneously responding to the challenge of the question and arriving at answers subconsciously. Okay, that is what you are going through. This is an evolution. You are in the cradle of evolution, intellectual evolution. All right. You understand this? This reaction you understood? Next reaction is, what do we have now? 1,3-bisphosphoglycerate. You see what happens next. The very next reaction, you are again breaking it down. 1,3-bisphosphoglycerate is converted to 3-phosphoglycerate. You had glyceraldehyde 3-phosphate. You removed some hydrogen, attached to phosphate group, converted into 1,3-bisphosphoglycerate. Then you are again removing phosphate group from that and converting it into 3-phosphoglycerate. By this, what did you do? Here you are using kinase because you are removing phosphate from the substrate. When you are removing phosphate from the substrate, when you are breaking it off, phosphate is being removed, energy is released when you are breaking the bond. That energy is used for giving this phosphate to who? Who is getting this phosphate? Who gets this phosphate? ADP gets the phosphate and it becomes ATP. So this is an ATP producing reaction. Reaction number 7 is ATP producing reaction. Reaction number 5 is 6 is NADPH producing reaction. Alright. So this one 7, phosphoglycerate kinase is the enzyme. Why the name is very clear. It is a phosphoglycerate that is being acted upon by breaking off phosphate and transferring the phosphate to ATP. So the nature of the enzyme is of kinase. It's a phosphoglycerate kinase. 3-phosphoglycerate is converted to 2-phosphoglycerate. What is mutation? Mutation is alteration of something. The primary thing remains the same, but there is alteration of something. What is being mutated here? 3-phosphoglycerate becoming 2-phosphoglycerate. Phosphate is taken from the third group, third position, and shifted to the second carbon. From the third carbon, phosphate is being shifted to second carbon. So the nature of the enzyme is that which brings about mutation, alteration of the structure of the same compound. Alteration of the structure of the same compound. So the enzyme is mutase. What mutase? You have phosphoglycerate which is being mutated. The enzyme is phosphoglycerate mutase. Simple. 9th one, phosphoglycerate, 2-phosphoglycerate is being converted into phosphoenol pyruvate. Phosphoenol pyruvate. So you are changing the nature of the compound and you are making it an enol compound. Enzyme is enolase. That's all. And phosphoenol pyruvate remove the phosphate from it, it becomes pyruvate. Enzyme is pyruvate kinase. So to convert one glucose into 2 pyruvate, step alteration. Keep changing, that's all. Alright. It's like Rubik's Cube. When you are solving Rubik's Cube, you have all the colors in the cube. Do you pluck it and paste it all around? You keep changing it, and at the end of it, you have all the sides done properly. Alright. Now, I will tell you the substrate. Okay, we'll go this way. I'll tell you the substrate. You have to tell me what it gets converted to, which is the enzyme that is doing it. Let's try. You should not look at the board. Alright. Glucose becomes glucose 6 phosphate. Enzyme hexokinase. Glucose 6 phosphate becomes fructose 6 phosphate. Enzyme phosphohexoisomerase. Phosphohexoisomerase or phosphoglucoisomerase. Okay, fructose 6 phosphate becomes 1,6 fructose 1,6 bisphosphine. Enzyme phosphofructokinase. Don't look at the board. No matter what the temptation, don't look at the board. Phosphofructokinase. Fructose 1,6 bisphosphate becomes what? 6 carbon is becoming into 2 3 carbon compounds. Both are aldehydes. Glyceraldehyde 3 phosphate and dihydroxyacetone phosphate. Reaction number 4. Reaction number 5. Isomerase reaction. What gets isomerized into what? Dihydroxyacetone phosphate becomes glyceraldehyde 3 phosphate. Enzyme phosphoglyceroisomerase. That's all, no? Phosphoglyceroisomerase. It's a phosphorylated glycerol compound being isomerized into something else. Then glyceraldehyde 3 phosphate becomes phosphorylation plus dehydroxylation. Dehydrogenation. Enzyme is dehydrogenase. So what will it become? Glyceraldehyde 3 phosphate becomes 1,3 bisphosphoglycerate. 1,3 bisphosphoglycerate. Enzyme is phosphoglycerate dehydrogenase. Phosphoglycerate dehydrogenase. Now 1,3 bisphosphoglycerate becomes 3 phosphoglycerase. Enzyme phosphoglycerate kinase. 3 phosphoglycerate becomes 2 phosphoglycerate. Enzyme is phosphoglycerate mutase. 2 phosphoglycerate becomes phosphoenol pyruvate. Enzyme is enolase. Phosphoenol pyruvate becomes pyruvate. Enzyme is pyruvate kinase. Alright. You may not remember the terms very clearly yet because you have not revised it repeatedly. But is the process clear? Do you understand it now? Transformation, stage by stage transformation. Okay, I will tell you the enzyme. I'll tell you the enzyme. You have to tell me the reaction for it in glycolysis. Alright? That will make it easier. All this to make you remember these names. Process you remember. Glucose to glucose 6 phosphate. Phosphohexoisomerase. Come on, Phosphohexoisomerase. Glucose 6 phosphate 2. Don't look at the board. Fructose 6 phosphate. Phosphofructokinase. Fructose 6 phosphate 2. Fructose 1,6 bisphosphate. Aldolase. Fructose 1,6 bisphosphate 2. Glyceraldehyde 3 phosphate and dihydroxyacetone phosphate. Next, phosphoglyceroisomerase. What to what? Dihydroxyacetone phosphate to glycerol phosphate. Next, phosphoglycerodehydrogenase. Glyceraldehyde 3 phosphate to 1,3 bisphosphoglycerate. Next is kinase. What kinase? Phosphoglycerokinase. 1,3 bisphosphoglycerate to 3 phosphoglycerate. Next is 3 enzyme. Phosphoglyceromutase. 3 phosphoglycerate to 2 phosphoglycerate. Next is enolase, not aldolase. 2 phosphoglycerate to phosphoenol pyruvate. Next is pyruvate kinase. Phosphoenol pyruvate to pyruvate. I'll tell the enzyme. You have to tell me the energy balance sheet of that enzyme. Okay? What is produced and energy balance sheet. Hexokinase. ATP. ATP is produced or used? Used. Energy balance sheet? -1 ATP. Alright, okay. Phosphohexoisomerase. Energy 0. Phosphofructokinase. 1 ATP is used, energy balance sheet -1. Aldolase. Aldolase 0. Then phosphoglyceroisomerase. Energy 0. Phosphoglycerodehydrogenase. Phosphoglycerodehydrogenase. Always remember, when we talk of dehydrogenase as the enzyme, either NADH or FADH2 is produced. In Krebs cycle, out of the 8 reactions in Krebs cycle, 4 are dehydrogenase reactions. 3 are associated with NADH production, 1 is associated with FADH2 production. Easy to remember enzymes there. And which is a GTP producing reaction. 5 reactions are over. Then one is the one which produces citrate. So it is citrate synthase. 6 enzymes over. Easy to remember enzymes in Krebs cycle. Alright. Okay. Phosphoglycerodehydrogenase. What is produced? What is produced? Energy compound. NADH is produced. Energy balance sheet 3 ATP per NADH, 6 ATPs per molecule of glucose. Phosphoglycerokinase. ATP production or utilization, what? Phosphoglycerokinase. 1,3 bisphosphoglycerate to 3 phosphoglycerate. ATP is produced. Energy balance sheet +1/+2. +1 per reaction /+2 is per glucose molecule. Next enzyme, mutase. Energy balance sheet 0. Next enzyme, aldolase. Not aldolase, enolase. Energy 0. Next enzyme, pyruvate kinase. Energy +1 plus 2. That's all, no? I have problem remembering names and numbers, but I remember everything of these pathways. How? You have to build logic around it. All the things that I am telling you is logic. I can't remember names. I have spent 5.5 years with my MBBS classmates. I know about them. I know their story. I know what they are doing. I know about their family. I find it difficult to recall their names. So, in my phone, the contact name, when I meet somebody, it will be some 2-3 lines. Their name, how did I meet them, what do they do, what can I get back to them for. All of those will be part of the contact name. Fortunately, the smartphone allows us to write paragraphs in name. The earlier phones, the feature phones that we used to have, had limit in the number of characters that you can use for names. So smartphone is a savior for people like me. All right. So these things can be remembered. What we have to remember in scientific concepts, we can by looking at it from different perspectives. The same thing from different points of view. All right. Now again, I'll tell you the enzymes. You have to tell me reversible or irreversible. The reactions mediated by those enzymes are reversible or irreversible. Hexokinase, irreversible. Phosphohexoisomerase, reversible. Phosphofructokinase, irreversible. Aldolase, reversible. Phosphoglycerodehydrogenase, reversible. Phosphoglycerokinase, reversible. Phosphoglyceromutase, reversible. Enolase, reversible. Pyruvate kinase, irreversible. Do you see another question here? There are 4 kinases, all 4 of glycolysis. Name the 4 kinases that you see in glycolysis. Hexokinase, phosphofructokinase, then phosphoglycerokinase and pyruvate kinase. If these 4 are given, which one would you choose as the odd one? Phosphoglycerate kinase. Why? The reaction mediated by phosphoglycerate kinase in glycolysis is reversible. The other 3 enzymes are mediating irreversible reactions. Is the phosphoglycerate kinase, is it always reversible? Or are the other 3 enzymes always irreversible reactions? Reversibility of reactions is dependent on the concentration of substrate and product. If the substrate is more than the product, direction is this. If the product becomes more than the substrate, the enzyme will do reverse reaction. When we say reactions are irreversible, means product of those reactions are quickly removed. They are not allowed to accumulate. They may continue in the same pathway, or they may be utilized in multiple other pathways, making the product of that reaction not available in enough quantity for the enzyme to do the reverse reaction. You understand this? All right. So that's all. So glycolysis, we are done. We have looked at glycolysis in multiple perspectives in reasonable detail. All right. There are more ways of looking at glycolysis. We will do that when we get appropriate questions. Let us not do everything at the same time. We will do it when we get appropriate questions. This much of elaboration is necessary if we touch it. This is why I did not touch this glycolysis, Krebs cycle, electron transport chain in the previous cycle of discussions. Because if we touch it, this basic discussion needs to happen. And I wanted it to happen around a question. Now we have a question, we will do it. Further elaboration of this based on questions. All right. So, summary of net energy yield. Total ATP used, 2 ATPs. Reaction number 1 and 3. Name the enzymes. Hexokinase and phosphofructokinase. Good. Total ATP produced, 4 ATPs. Step 7 and 10. Enzymes. Enzyme number 7 is phosphoglycerate kinase. Step number 10 is pyruvate kinase. Total NADPH produced 2 NADH in step 6, which can yield approximately ATP, assuming 1 NADH is equal to enzyme. Step number 6, enzyme. What is the nature of the enzyme? If it is producing NADH, what kind of enzyme is that? It's a dehydrogenase. That you should be clear. Even if you don't know what should be preceding dehydrogenase, that's fine. You should not choose kinase for a reaction which is producing NADH. So be clear about the nature of enzymes. Here only we have got 4 or 5 different types of enzymes. Kinases we have. What are the different types of enzymes we have seen in this? You tell me. Kinase. Then go step by step. Aldolase. Before that we have isomerase. Kinase, isomerase, aldolase, enolase, mutase, dehydrogenase. 6 types of enzyme. Kinase will do what? Shift. What does it shift? Phosphate groups. Kinase will shift phosphate groups. Isomerase, interconvert isomers. Aldolase produces aldehydes. Mutase, what dehydrogenase? Pyruvate dehydrogenase. Pyruvate dehydrogenase. That's all. Why is it called complex? It is not just one, it is a combination of multiple chemicals which are a part of this complex, which will bring about this reaction. Coenzyme A is also a part of this complex. Alright? Pyruvate dehydrogenase complex is what converts pyruvate into acetyl-CoA. In this process, one carbon is removed from pyruvate and gets released as carbon dioxide. So for this, you need oxygen. This is oxygen dependent. Alright, this is oxygen dependent. Absence of oxygen will not allow pyruvate to get converted to acetyl-CoA. So in anaerobic conditions, pyruvate will go into lactic acid pathway, producing lactic acid, where it utilizes 1 NADPH. Pyruvate to lactate utilizes 1 NADPH. So glucose going in anaerobic pathway, net energy production is 2 ATP. Net energy production is 2 ATP because in glycolysis 2 ATP is produced, 2 NADPH is produced, 2 pyruvate is produced. Each pyruvate to become lactic acid utilizes 1 NADPH. So 2 pyruvates to get converted into 2 lactic acid, 2 NADPH is gone. What remains after the metabolism of glucose is 2 ATP. Look at the gross difference in the energy produced by aerobic metabolism and anaerobic metabolism. 38 ATP is produced by aerobic metabolism, 2 ATP is produced by anaerobic metabolism. Lactic acid accumulation in a tissue. Lactic acid accumulation leads to stimulation of, considering human beings. Lactic acid accumulation leads to stimulation of nerve endings which will interpret the accumulation of lactic acid as pain. Brain interprets lactic acid accumulation as pain. I'll give you an example. You don't have the habit of exercising, you are not used to running. But suddenly, you run a long distance. You will have cramps here. Correct? Your abdominal muscles and your diaphragm will start cramping up. Why is that happening? Your muscles have gone into a hyperactivity mode. Hyperactivity mode demands more supply of nutrients, demands more supply of glucose for energy production, demands more oxygen supply for oxidative metabolism to happen in the mitochondria, so that glucose breakdown leads to ATPs, which will be used by the muscle for activity. For the supply of glucose and the supply of oxygen, the dependency is on circulation. The supply of blood to the working tissue should be matching the intensity of the work the tissue is carrying on. The muscle is working very hard, it is going into a hyperactivity mode. The blood supply should match it. But if you are not trained, your heart is not capable of pumping so much. Your heart is not capable of pumping so high volumes of blood in every cardiac cycle. The cardiac output will not be so high. The number of heartbeats, there is a limit to how much a heart can go. So there is a mismatch in the availability of oxygen for glucose to be taken in an aerobic pathway. But ATP needs to be produced for muscle to work. So what happens, to the extent of availability of glucose, availability of oxygen, glucose is being metabolized in Krebs cycle, ATP is produced in electron transport chain. But more glucose is being broken down. Glycolysis is happening. Glycolysis is not dependent on oxygen. So glycolysis matches the rate of muscle activity. Glucose is being broken down into pyruvate. That is going on. You see, pyruvate kinase activity is reversible or irreversible? Irreversible. So pyruvate cannot become phosphoenol pyruvate and go back in the pathway. So whatever pyruvate is produced is there. It has to be utilized. If oxygen is available, to the extent of oxygen being available, pyruvate is going into pyruvate dehydrogenase complex, gets into Krebs cycle, ATP is being produced. But there is still more pyruvate available. It will not be allowed to stay there. Pyruvate will be taken up by lactate dehydrogenase. Pyruvate will be converted into lactic acid. You are understanding this? So, on one side, glucose is being broken down in aerobic respiration pathway and it is converted to carbon dioxide. We breathe out. We are breathing heavily, we breathe out. But because we are not able to supply oxygen to match the demand of the working muscle, some of the pyruvate is also being converted into lactic acid. When the accumulation of lactic acid reaches a threshold where the nerve endings are picking it up and sending the signal to the brain telling that the lactic acid has accumulated. Accumulation of lactic acid beyond a certain threshold is toxic to the cell. It will damage the cell. Then the brain will start sending signals of pain. The brain will start interpreting the accumulated lactic acid in the form of pain. So the area where lactic acid is accumulated, the brain will project pain there. We will experience pain. And when you experience pain, what do? Will you still continue to run? You will slow down. I can't run any further. I can't run. You will stop you will reduce the intensity of activity. You will more likely stop. You will sit down. When you sit down, the demand has reduced. The supply of oxygen increases. Whatever pyruvate is still pending, accumulated to be taken up, will be taken up more into aerobic pathway. Accumulated lactic acid will slowly start getting cleared. And when the lactic acid comes below the threshold where it is being interpreted as pain, you will experience the absence of pain. Pain will reduce and you will feel better. Pain is a protective mechanism. Alright, so you understand this? Why this is important? Oxygen dependency is at 2 levels for our body. During high intensity working, for metabolism to go into aerobic pathway, for glucose to be broken down in an aerobic manner, oxygen is required here also in the pyruvate dehydrogenase complex, and oxygen is required as the terminal electron acceptor in the electron transport chain. 2 places we need oxygen. Alright. This is what happens in the nursery classrooms of our school. One kid tells, "Oh, so I need to go to the washroom," and suddenly there is a train of children going one behind the other. All of you should plan your bladder management properly. All right, 9:25 should be your 9:20 to 9:30 should be your bladder emptying time in your room. All right, so that 9:30 to 12:30 will be filling time. Okay. Okay, we'll go down further. Good. I thought we will not be able to complete all the three in one session. We will complete. Go. All right. So here in oxidative decarboxylation, what enters? Pyruvate. What exits? Acetyl-CoA, which enters the Krebs cycle, and carbon dioxide as a byproduct. Enzyme pyruvate dehydrogenase complex, a multi-enzyme complex that catalyzes this conversion. It is not a single enzyme, it is a multi-enzyme complex. What? Not yet. Just remember it as a multi-enzyme complex. Alright, we will not break it down further. We will break it down further when a question comes on this. Today let's get the most critical aspects of these things. Reversibility, irreversible under normal cellular conditions. Energy used, no direct ATP is used in this reaction. Energy produced, 1 NADH is produced per pyruvate molecule. Net energy 3 ATP in equivalent. So per glucose, oxidative decarboxylation contributes to 6 ATP molecules. Energy equivalent to 6 ATP molecules. Alright. This conversion takes place in the mitochondria where pyruvate is decarboxylated and the resulting acetyl group is attached to coenzyme A to form acetyl coenzyme A. Alright. So there is decarboxylation that needs to happen. Decarboxylation needs to happen. Dehydrogenation needs to happen. And coenzyme needs to be acetylated. So if you look at it, there are 3 reactions that are happening here, separate reactions. Enzymes for all this should be there in the complex. This is a crucial step linking glycolysis to the Krebs cycle. This is the context. Citric acid cycle. How many reactions are there in citric acid cycle? We have 8 reactions. Same format. What enters, what exists, enzyme, reversibility, energy used, energy produced, net energy. Alright. We will look at it in terms of number of carbons also. Acetyl-CoA and oxaloacetate. Oxaloacetate is a how many carbon compound? 4 carbon compound. Acetyl-CoA is 2 carbon compound. They combine to produce what? Citrate or citric acid, which is a 6 carbon compound. Here I want you to remember the number of carbons. In glycolysis it is clear. 1, 2, 3, you have 6 carbon compounds. Number 4 will give you 2, 3 carbon compounds. Beyond that, all are 3 carbon compounds. So there will not be questions that will lead you to confusion there. Here it will confuse. Citrate is 6 carbon, oxaloacetate is 4 carbon, acetyl-CoA is 2 carbon. So we are starting with a stable compound, citrate. Enzyme is, I told you, it is an enzyme which is combining 2 things to produce citrate. So the enzyme is synthesizing citrate. Therefore it is named as citrate synthase. Energy, nothing. Alright. Reversibility, irreversible. So acetyl-CoA, once it enters into the Krebs cycle, it cannot go back. The intermediate compounds can go into different pathways, that's okay. But acetyl-CoA cannot exit. You get in, you have to go through the journey. Alright. You are all like acetyl-CoA in the Krebs cycle of Star 30 campus. You can't get out of here. Alright. So eventually metabolism should happen, energy production should happen, whatever happens we will see. Transformation, evolution. Okay. Anybody feeling the process like a torture? No, not yet. So maybe I have to increase the intensity of torture. Don't feel it like torture because there is a lot of things going on. You have exercise every day, you can play every day, you have time to make friends, spend time with friends, you have time for movie. It's a very balanced program. Torture might be because you don't have access to your phone. Anybody still feeling the separation anxiety because of being separated from your phone? Or that that separation pangs. You still get that? You still wake up in the night searching for your phone on the bed? It's still happening? Okay. You will forget. It's like losing somebody very near and dear, losing a good friend. You will keep remembering the friend for a long time, for some time, and eventually you will start to live without the friend. Alright? So second reaction is citrate becomes isocitrate. The enzyme is aconitase. The enzyme is aconitase, reversible. See, we look at the irreversible reactions, okay? The first reaction where citrate is being produced is irreversible. Isocitrate dehydrogenase, where NADH is being produced, irreversible. Alpha ketoglutarate, where NADH is being produced, irreversible. Notice here, it is not only NADH being produced. These are the two reactions where carbon dioxide is being released or removed. Carbon is removed in the form of carbon dioxide. So naturally you want it to be irreversible. You don't want carbon dioxide entering through this and forming compounds in the system. So these are the two reactions which are irreversible here. Again, next reaction, ATP production reaction. It is reversible, 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 reversible. How many irreversible reactions are there? 3 irreversible reactions. What are the 3 irreversible reactions? The acetyl-CoA entry reaction, carbon exit reaction. So can I say the reactions where carbon is entering and exiting Krebs cycle are irreversible? Yes or no? In the form of acetyl-CoA, what are you bringing into the system? You are bringing in 2 carbons, which you are supposed to remove. And in these 2 reactions, you are removing 1 carbon each. So the reactions where carbon enters in the form of acetyl-CoA and carbon exits in the form of carbon dioxide are irreversible. All other reactions of citric acid cycle are reversible. So these 3 reactions, one is the reaction, entry reaction is a citrate synthesizing reaction. So the enzyme is citrate synthase. Carbon exit reactions are NADH producing reactions. So the reactions are mediated by dehydrogenase. The reactions are mediated by dehydrogenases. Alright. There is one more dehydrogenase, FADH producing dehydrogenase. Clear? Now we will look at it. We will look at the energy reactions only. Isocitrate is being converted into alpha ketoglutarate. See here, how many carbons? 6 carbons. Isocitrate, it is also an isomeric form of citrate. It is also a 6 carbon compound. Isocitrate being converted into alpha ketoglutarate. 1 carbon is removed. Alpha ketoglutarate is 5 carbon compound. Alpha ketoglutarate gets converted into succinyl coenzyme A. Succinyl CoA. 1 more carbon is removed. So this is 4 carbon compound. Of everything that is there in Krebs cycle, except that which enters. That which enters is acetyl-CoA, 2 carbon compound. Citric acid is 6 carbon, alpha ketoglutarate is 5 carbon. Everything else is 4 carbon. What are those everything else? Succinate, fumarate, malate, oxaloacetate. That's all, no? If you see, oxaloacetate is entering in the first reaction, oxaloacetate is exiting the last reaction. Correct? Cycle, no? You bend this now. What exits in the last reaction enters in the first reaction. For something to be a cycle, The starting point and the end point should be the same. Starting point is oxaloacetate, end point is oxaloacetate. To oxaloacetate you are adding acetyl-CoA, becomes citrate. Citrate becomes isocitrate. Isocitrate becomes alpha ketoglutarate. Alpha ketoglutarate becomes succinyl-CoA. Then it becomes succinate, fumarate, malate, oxaloacetate. Alright. Citrate you can remember. Citrate you can remember citric acid because it's named after citrate, citric acid. Alpha ketoglutarate you can remember. Unique name, remember alpha ketoglutarate. After that, what is the entry and exit compound? Oxaloacetate. In between you have to remember 3. In between you have to remember 3. Do you all listen to radio? There is Red FM. We have here SFM. Succinate, fumarate, malate. That's all. Huh? We have Red FM, no? SFM. I need to figure out ways to remember sequences. Otherwise I can't. So there is a saying, if you want the shortest possible or the most efficient way of doing a task, give the task to a lazy person. He will figure out ways to make it easier. Alright. So a person who can't remember sequences, numbers and names has to remember it. So I have to come up with ways of remembering it. Now we will see. We will list down the compounds. What enters? What is the starting compound? Oxaloacetate. Then converts to citrate. Then isocitrate. Then alpha ketoglutarate. Then succinyl-CoA. Then succinate, fumarate, malate, oxaloacetate. Over. Easier than glycolysis, no? Alright. So, where is the, we will look at it again one by one. Look at all the dehydrogenase reactions. Isocitrate to alpha ketoglutarate, alpha ketoglutarate to succinyl-CoA, and succinate to fumarate. These are the 3. If you look at the energy reactions, just look at it here. Isocitrate to alpha ketoglutarate, that is the third reaction, 3, 4, 5, 6. All 4 are energy reactions. 3, 4, 5, 6 are energy reactions. 7 is not an energy reaction. 8 is an energy reaction. You notice that? 3, 4, 5, 6, 8 are energy reactions. 3, 4, 5, 6, 8 are energy reactions. 3, 4 are NADH producing reactions. 5 is GTP reaction. 6 is FAD. 8 is NAD. Clear? So 3, 4, 6, 8 are dehydrogenase reactions. 3, 4, 6, 8 are dehydrogenase reactions. 5 is a GTP producing reaction. So it should be a kinase reaction. There is the kinase there. Succinyl-CoA synthetase, or it can also be a succinate kinase. You understand? The enzyme can be succinate kinase also. Succinyl-CoA synthetase or succinate kinase. Why succinyl-CoA synthetase? Why can't we call it succinate synthetase? We can call it. It's a reversible reaction. You can call it succinyl-CoA synthetase or succinate synthetase or succinate kinase. It means the same. Questions can come with any of the 3 names. It is not necessary that this name only will come. When you know the nature of the enzyme, what it is doing and what is it acting upon, it's easy to remember. You can easily interpret it and arrive at the answer. Alright. So Entry of carbon, reaction number 1. Exit of carbon, reaction number 3, 4. Reaction number 3, 4 is exit of carbon. Reaction number 5 is GTP synthesis. Reaction number 6 is NADH, FADH. Reaction number 8 is NADH. Now, we have listed down the sequence of compounds. We will list it down in terms of what becomes what, reaction number wise. Reaction number 1, forget about what enters and what exits. We will talk about only those that are circulating in the cycle. Reaction number 1, what becomes what? Oxaloacetate becomes citrate. Reaction number 2, citrate becomes isocitrate. Reaction number 3, isocitrate becomes alpha ketoglutarate. Reaction number 4, alpha ketoglutarate becomes succinyl CoA. Reaction number 5, succinyl CoA becomes succinate. Number 6, succinate becomes fumarate. 7, fumarate becomes malate. 8, malate becomes oxaloacetate. Okay. 5, 6, 7 you will not forget, no? What after what you cannot forget, SFM. Alright. Now we have mentioned this. We will list down the enzymes in a sequence. Enzyme number 1, it is synthesizing citrate. So it is citrate synthase. Then enzyme number 2, citrate is becoming isocitrate. The enzyme is called cis-aconitase. It can be an isomerase, cis-aconitase, changing the form. Okay, remember that. Aconitase is the second enzyme. Question will not come on that enzyme. Okay, you can skip that step also. Citrate to isocitrate, isocitrate to alpha ketoglutarate. It's just a transitional form. Citrate to isocitrate is a transition state. Citrate is getting converted to alpha ketoglutarate. In between you have a transition state. Remember aconitase. Isocitrate to alpha ketoglutarase. The enzyme should be, I told you, this is a energy producing reaction, NADH producing reaction. So the nature of enzyme is dehydrogenase. What is it acting on? The enzyme is acting on reaction number 3. The enzyme is acting on isocitrate. So the name of the enzyme should be isocitrate dehydrogenase. What is it producing? Isocitrate dehydrogenase is acting on isocitrate and producing alpha ketoglutarate. Reaction number 4, alpha ketoglutarate is being acted upon by enzyme. It is also a dehydrogenase reaction and produces succinyl CoA. So enzyme is alpha ketoglutarate dehydrogenase. That's all. Alpha ketoglutarate dehydrogenase. Done. Alpha ketoglutarate is converted to succinyl CoA. Next, succinyl CoA should get converted to succinate. Enzyme is succinate synthase. Succinyl CoA synthase or succinate kinase. It is also an energy reaction. Reaction number 5 is also an energy reaction. What is it producing? GTP. Alright. Next is succinate becomes fumarate. Again an energy reaction, a reaction which will produce FADH2. Succinate becoming fumarate. Energy reaction, FADH2 is being produced. So the nature of enzyme should be dehydrogenase. What is it acting on? Succinate. So the name of the enzyme is succinate dehydrogenase. Fumarase is produced. Now fumarate is getting converted into malate. No energy. The enzyme is fumarase. Fumarate, something is being broken down there. Alright, we'll see that in the later stage. So the enzyme is acting on fumarate to convert it into malate. Enzyme is simple, fumarase. Last one is malate is getting converted into oxaloacetate. It is again an energy producing reaction. NADH is being produced. Nature of enzyme should be dehydrogenase. What dehydrogenase? Malate dehydrogenase. That's all. Over. Easy. Can you tell me the enzymes which mediate the 4 energy reactions of Krebs cycle? From the top. Don't look at the board. Enzymes which mediate the energy reactions of Krebs cycle. Just visualize. Take your time. Isocitrate dehydrogenase, first one. Very good. Then alpha ketoglutarate dehydrogenase. Then succinyl CoA synthase or succinate synthase or succinyl kinase 3. Okay. Then alpha ketoglutarate dehydrogenase, succinate dehydrogenase, malate dehydrogenase. Succinate dehydrogenase. Why? Succinate dehydrogenase doesn't produce GTP. It produces FADH2. The other 3 produces NADPH or NADH. Alright. Now, let us recall all the enzymes starting energy producing enzymes or energy producing enzymes starting from glycolysis to Krebs cycle in a sequence. Alright. Let us try. Or let us tell the compounds in a sequence. Let us tell the compounds in a sequence, not the reactions. We will just tell the compounds in a sequence. Glucose. Next is glucose 6-phosphate. Fructose 6-phosphate. Fructose 1,6-bisphosphate. Glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. Then glyceraldehyde 3-phosphate. This gets converted to this. Then 1,3-bisphosphoglycerate. Then 3-phosphoglycerate. Then 2-phosphoglycerate. Then phosphoenol pyruvate. Then after pyruvate, this is pyruvate now. Acetyl CoA, what is this? Carbon dioxide. So what we have? Acetyl CoA. Acetyl CoA what is this? This is oxaloacetate. Citrate. Then isocitrate. Alpha ketoglutarate. Succinyl CoA. Then succinate, fumarate, malate, oxaloacetate. So you don't even have to write reactions like this. You can just write the sequence of compounds. Okay, we'll see enzymes now. Enzymes. Alright. Glucose to glucose 6-phosphate. Hexokinase. To fructose 6-phosphate. Phosphohexo isomerase. Fructose 1,6-bisphosphate. Phosphofructokinase. G3P and DHAP. Aldolase. DHAP to G3P. Phosphoglycero isomerase. G3P to 1,3-BPG. Glyceraldehyde 3-phosphate to 1,3-bisphosphoglycerate. Phosphoglycero dehydrogenase. Then 1,3-BPG to 3-PG. 1,3-bisphosphoglycerate to 3-phosphoglycerate. What is the nature of the enzyme here? It has to be a kinase. What kinase? Phosphoglycerate kinase. Then 3-phosphoglycerate to 2-phosphoglycerate. Mutase. What mutase? Phosphoglyceromutase. 2-phosphoglycerate to, what happens next? Phosphoenol pyruvate. Enzyme is enolase. Phosphoenol pyruvate to pyruvate. Enzyme is pyruvate kinase. Pyruvate to succinyl CoA, acetyl CoA. Enzyme is pyruvate dehydrogenase complex. Then acetyl CoA plus oxaloacetate to citrate. Enzyme citrate synthase. Citrate to isocitrate. Aconitase. Isocitrate to alpha ketoglutarate. Isocitrate dehydrogenase. Alpha ketoglutarate to succinyl CoA. Alpha ketoglutarate dehydrogenase. Succinyl CoA to succinate. Succinate kinase or succinyl CoA synthase. Succinate to fumarate. Succinate dehydrogenase. Fumarate to malate. Fumarase. Malate to oxaloacetate. Malate dehydrogenase. Oh, done, no? See how easy it is. Electron transport chain, we have already seen the story. We will elaborate the story and get the complexes and all a little bit more in the next session. All right. What we have discussed today is the basis for understanding photosynthesis also. Reactions, whenever you learn pathways, remember this, in your first year MBBS biochemistry, you will have a lot of pathways to remember. If you try to remember it like this, arrow marks, you will lose out. For any pathway, serial number, what enters, what exists, what is the enzyme, reversibility, energy. Energy in, energy out, balance sheet. If you write it like this, it will start appearing like a picture in front of you. So what I have trained you today is not only helpful for understanding respiration and photosynthesis during your NEET preparation, it will be helpful for you to do your biochemistry better during your first year MBBS. All right. Okay. We will just wait. We still have 2 more minutes. All right. Okay, this is done. Pyruvate dehydrogenase. Then this we discussed over. We'll just look at the summary. Total ATP produced per turn, per cycle. Per cycle, talk about it. Total ATP, 1 as GTP. Total NADH per cycle, 3. Total FADH2, 1. Total energy ATP equivalent per cycle, 12. 3 into 3, 9 plus 2 plus 1. So per molecule of glucose, this happens twice. So per molecule of glucose, energy in ATP equivalent produced via Krebs cycle is 24. So 8 ATPs from glycolysis, 6 ATPs from oxidative decarboxylation, 24 ATPs from Krebs cycle. Summary per glucose molecule, 24 ATPs. Each step is again explained in words also. Then comes our master, electron transport chain. Okay, we will look at it in the next session tomorrow. Okay, feeling good? Knowledge, the power of knowledge is that there is a saying of my master Swami Vivekananda about education, what education is. Education is the manifestation of the perfection that is already in man. All of us know all of this. We are a part of all of this. No, it's all happening in us. That means our body knows it. When we understand it, it is not understanding coming from outside. It is a realization that is coming from inside. So you are realizing something that is happening inside you. You are realizing something that you are a part of. All right. So that's a different kind of kick. It gives you a kind of happiness from inside when you understand things, when you realize things. All right. So you should always try to experience the knowledge. You should try to get it. When you get that feeling, no, you will have a kind of cold, tingling sensation that happens inside your body when you get that realization of knowledge. All right. So I always tell my teachers when I train teachers, 2 things should happen in a classroom regularly. There should be a lot of aha moments. Oh, is this it? Oh, when you think, when you are made to think, when there is a lot of thought going on in trying to get answers, when suddenly the answer strikes, like when you told who is the electron donor for complex 1, who is the electron donor for complex 2, why will we die if there is no oxygen? Oxygen is the electron acceptor. That is the aha moment. Aha. There should be aha moments in class. That is a good class. If a teacher is not giving enough aha moments in the class, it's a boring class. And there will also be, if students are pushed into thinking, there will also be ayyo moment. What is that ayyo moment? You think about something so much, you don't get an answer to it. But when you know what the answer is, I could have got it. All right. Those IIO moments or IO moments will make you will make your mind open up thinking process. The aha moments will make your mind get excited about learning. If these 2 are not there in every class, in every learning process, learning is not happening. Simple data transformation is attempted. Data cannot be transferred. As data is being delivered, it has to transform into knowledge. Then it will go into you. And when that knowledge somewhere gives you a glimpse of wisdom, then you will have that aha moment. Thinking will give you ayyo moments. Okay, all the best. We'll meet in the next class tomorrow. Any questions, any doubts? I'm here. Ask. Okay, we'll close for the day.